Bismillahirrahmanirrahim. We start another chapter. We have started the chapter, but this is the second video on this chapter, video 2 on inheritance, and we're going to be covering 17.3. This is for O-level biology in the syllabus 2023, which we are covering. Bismillahirrahmanirrahim. We start every session, everything with Bismillahirrahmanirrahim. Now, as we come to 17.3, it is describe inheritance as the transmission of genetic information from generation to generation. Now, how does that happen? We're going to discuss that. Then define an allele as an alternative form of a gene. Understand and use the terms dominant, recessive, phenotype, genotype, homozygous, heterozygous. Use genetic diagrams including Punnett squares to predict the results, 1:1 and 3:1. Explain why observed. This is a very important point, and I'm going to take some quite a bit of a time of yours to explain this to you all. Why observed ratios differ from expected ratios when there are small numbers of offsprings. State two identical homozygous individuals that breed together will be pure breeding. Pure breeding means those will be homozygous. Either they will be homozygous dominant or they will be homozygous recessive. Then explain codominance reference to AB or blood groups because AB is a codominant phenomena. Then determination of sex in humans. Why is there a male offspring and why is there a female offspring, and what is the difference in the male and the female genetics. Then describe a gene mutation. Then describe a chromosome mutation. So there are two different types of mutation: gene mutation and chromosome mutation. And this is of course new in the syllabus. So state that mutation, meiosis, random mating, random fertilization are sources of genetic variation. Understand that ionizing radiation and some chemicals increase the rate of mutation. So chemicals like tar and cigarette smoke, so all those who smoke, they increase the risk of mutation, which results in lung cancer. And ionizing radiation, if you expose too much to UV light, you go sunbathing, then the person can get skin cancer. Now, basically, the first point of the syllabus is inheritance. Inheritance means what? It means the transmission of genetic information, generation to generation. Now, how? How? Basically, every cell has a nucleus. And inside the nucleus, you have chromosomes. And chromosomes are made up of DNA. And then we have the genes. So the genes make up the total genetic information, which is also called the genome. Now, how is that passed on from one generation to the other? Now, you've got to understand is the sperm fuses with the ovum. So there is one of each pair of chromosomes coming from the sperm nucleus and one coming from the ovum nucleus. Now, when this fertilization takes place, what is formed? One cell, the zygote. The zygote contains genetic information from the mother cell and genetic information from the male, the father cell. Now, both this genetic information now passes on to the new cell, which is called the zygote. Now, this zygote is going to develop into another human being or an organism or any other living organism. So now the genetic information from one generation, this generation, has passed on to the second generation. So this is the first generation and this is the second generation. So genetic information, now you've got to understand it instead of just remembering a transmission of genetic information generation to generation. So the sperm and the ovum, so this would be the sperm, this would be the ovum, that would fuse to form the zygote. Now from the zygote develops another human being. So these are the first human beings, and then now this is another generation. So the children from that couple will now have the genetic information from the parents. Now let's look at another important point of the syllabus. It's what is an allele. An allele is an alternative, allele word, the first alternative form of a gene. Just like I have markers. So I have a green marker and a red marker when I'm writing on the board. Or I have a yellow marker and a black marker and a gray. So alternative form of a gene. Now I'll give you a simple example that the color of fur in rabbits can either be black or white. Just giving you an example. Now we decide as biologists to give it a letter. And I say, okay, let's say it's big B is for black fur and small b is for white fur. Now, we as biologists will decide that because we have to know when we give it a capital letter, like big B. Now that means this is a dominant trait. That means this is a dominant trait. If I've said in the question it's a capital letter, then it's a dominant trait. But if I've given it a small letter, that means this is a recessive trait. Now, what is dominant and recessive? Dominant means that which is going to show its effect. Recessive means that only if both of them are recessive, that means if it is homozygous recessive, both are small b, only then in an organism, in a rabbit, will that be white fur. So the black furred rabbits can either be big B or big B, small b. Now, what does this mean? Big B means homozygous dominant. Big B, small b means heterozygous. And small b means homozygous but recessive. So now this is called the genotype. This is called the genotype, which is in the form of letters. So genotype is always either big B, or big A, or big D. You can write it anything. It will of course be given in the question. What are the letters that you're going to use? That is, the Institute of Biology decides those letters for each specific traits. But then what is this going to be? We say big B are black furred rabbits. So this will be the phenotype. Phenotype means physical characteristic, PH. Physical features. Phenotype is the physical features. You can look at a rabbit and say it's got black fur, or it's got black fur, or it's got white fur. So you have to know the difference between phenotype, and I've just said phenotype is physical features, which actually you can see that this rabbit is black furred or white furred. So that is a phenotype. Genotype is when I say letters. When I say letters like big B, or I can say big A, or I can say big D, or I can say big A, or I can say small a. Now this is all genotype. So genotype is always written with using letters A, B, D. We don't use the same one. Like C, big C and small c is the same, so we don't like using these sort of letters. Now coming to the monohybrid cross. This is a monohybrid cross. Mono means one, so you're only discussing one phenotype. You're only discussing the color of the petals. So they can be either purple or white. Now, when do you get a 3:1? This is the important thing that you have to understand. 3:1 you only get when one parent, which is heterozygous, is crossed with another parent which is heterozygous. Now, there are three which will be, so 75% of the flowers will be purple and 25% will be white. But what will be, so the phenotype ratio is 3:1. The phenotype ratio is 3:1. Why? Because 1, 2, 3 purple and 1 white. So the phenotype ratio is 3:1. But what will be the genotype ratio? The genotype ratio will be 1:2:1. Why? This is 1, then these are the 2, and then 1. So the genotype ratio will be 1:2:1. But the phenotype ratio will be 3:1. So the phenotype means physical features, means what can you see? You can see the purple colors. You can see the white color. So there are three times the purple flowers and one as compared to that one. So if there are 30 purple flowers, there will only be 10 white flowers, right? So if there are 30, so I can just do another thing like this: 60, 20, 120, 40. So this is 3:1. This is 3:1. Now let's look at another word that we use very frequently, and that is called pure breeding. Pure breeding means what? What are pure breeding organisms? Pure breeding, we only use the word when there is only one type of gamete. One type of gamete means either the pure breeding ones will be this, or the pure breeding one will be this, or they will be big B. So the gametes that they produce now, so if we have the parents are big B, pure breeding, and is crossed with a homozygous recessive, now all the offsprings will be heterozygous. So we've crossed a blue flower with a white flower. Now all the offsprings are blue. So when you cross a homozygous dominant with a homozygous recessive, this is heterozygous. Now this is called the F1 generation. This is called the F1 generation. But when you cross the two heterozygous, which is the F1 generation, then this what you get here is the F2 generation. So the 3:1 is in the F2 generation. Now I want to talk about the codominant phenomena. Codominant phenomena now, for instance, if you look at all my previous examples, I said black furred rabbit into white, all black. So black is dominant. But if I say black into white, all offsprings white, then that means white is dominant. But here I come up with three phenotypes. I said red flowers are crossed with white flowers and all the flowers are pink. So red into white, all pink. So we have three phenotypes. When we have three phenotypes, it is called codominant phenomena. But if I had said red into white, all red, then red dominant. If I had said red into white, all white, white dominant. But now I'm saying red into white and all the offsprings are pink. Now this is a phenomena of codominance. So you use two letters. You use two letters, C raised to the power R. So the red will be C raised to the power C raised to the power R, and the white will be C raised to the power W, C raised to the power W. Now there won't be a big R and a small r, because we give it another letter, CR, or we say CW. So CR, CW are pink. Now if you cross two pink flowers, now some we get red, some we get pink, and some we get white. Now this is 1:2:1. The phenotype is also 1:2:1, and the genotype is also 1:2:1. So this is a phenomena of codominance. Now looking at the alleles for blood group, we have three alleles for blood group. Now very basic for you to understand. We have IA, we have IB. So how did we find this out? We looked at the blood groups of 10,000 people, and we realized that there were only three alleles. But then we have four blood groups. Well, how do we have four blood groups? Well, we have number 1, number 2, number 3, number 4, number 5, number 6. We have 6 genotypes. But we only have 4 phenotypes. Phenotypes are the one which we can check or we can see. So this is 1, this is 2, this is 3, and this is 4. So we have 4 blood groups. So that means we have 4 phenotypes. Now this is a typical MCQ which they give you on this. But then we have 6 genotypes. By 6 genotypes, I mean 1, 2, 3, 4, 5, and 6. 6 genotypes. So these are the alleles for blood group. Please remember that whenever we do genetics questions, you're going to do it per line. It's going to be marked per line. The first line is parental phenotype. The second line is parental genotype. The third line is gametes. Number 4th is offspring genotype, and number 5th is offspring phenotype. Now I will show you how you need not write these headings, but you need to do the questions in this manner under these 5 headings. So number 1, like for instance, we say there is a couple and one of them is blood group A and one of them is blood group B. So that will be the phenotype. Now what will be the genotype? Now the genotype can be it can be a lot of things, but I'm just giving you something very basic. So parental genotype IA, and the parental genotype of the B group person, whether it's the mother or the father. So this will be IB. Okay, now how do we write the gametes? Very easy. How do we write the gametes? We just write them separately. The same line, this one you write here, and you write this a little distance away from it, and you circle it. This means that these are the gametes. Like if this is the male, the blood group A person is the male, so the sperms will either have IA or IA, so there's not difference in that. Similarly, here also we write the same. IB, IB, because you write it just a little distance apart and you circle them. So this would be the line for gametes. Then offspring genotype. Offspring genotype means what would happen if, now I give you a very simple way how to do these crosses. 1, 2, 3, 4. So what if 1 and 3 fertilized? So then it would be IA, IB, right? What if 1 or 4 fertilized? So it is 1, 3, 1, 4, and then 2, 3, 2, 4. So then in this case, of course, all are IA, IB, so there is no difference. And then we have this one. This one is also IA, IB. And then you have the last one would be 2 and 4, which is also IA, IB. Now the offspring phenotype, what will be their phenotypes? They will be blood group AB. This one would be blood group AB. This one would be also blood group AB, and that one would also be blood group AB. So this is how you do the questions. I'm going to do another one so make you all understand this in a little better manner. Now if you look at this solved question, the parental phenotype, one of the parent was A group and the other was B group, right? So we have this A group and we have this parent B group. Now the genotype was IAIO. Now why did I give this genotype? Because in the previous one I didn't give this genotype. The reason is that the question said that how can a couple have 4 children with different blood groups? Now that is the only possible way that you could get 4 different blood groups. So if the A group parent was IAIO, so IAIO, and the B group parent was IBIO, so then the gametes will be IAIO written separately. I've just done that in blue. So you can see this one, and this one, and this one. So I've just written them separately and I've circled them. And then the offspring genotype, if the IAIO fertilize, if the IA sperm fertilizes the IO ovum, then we get an AB child. This will be the offspring genotype, and the blood group will be the offspring phenotype will be blood group AB. This will be IAIO blood group A. This will be IBIO blood group B. This will be IOIO blood group O. So now this means that there is a 25% chance. There is a 25% chance of having any of these blood groups. But they have 4 children and 4 of them are blood group O. Explain how. Or the question could be, this couple has 4 children, 2 are blood group A and 2 are blood group B. Now, how do you answer this question? The answer to this question is that there is a syllabus point. There is a syllabus point which you need to understand. The syllabus point is syllabus point 5. Explain why observed ratios differ from expected ratios, especially when there are small numbers of offsprings. Why is it that, they should have had, if there are 4 children, one should have been A, B, AB, and O. Why is it that 4 of them are blood group O? Or why are there 2 blood group O and 2 blood group A? Because 4 is a very small number. 4 is a very small number. You see, this couple has 4 children. That's a very small number. But if they had, say, 100 children, then this would roughly be the probability. That they would be 25% would be A, 25% would be B, 25% would be O, and nearly 25% would be AB. But because 4 children is a very small number, very number of offsprings is less. If this couple had many offsprings, then of course this number could have been maybe. So you study this in maths, I think, with probability. So the probability that each child would have their different blood group is only 25%, but they only have 4 children, or maybe have 2 children. So in 2 or 4 or 5 or 6, this probability does not stand true. So this is the part that you have to understand why observed ratios. Observed does not fit the expected. Expected was that if there are 4 children, one should have been AB, one should have been A, one should have been B, and one should have been O. But because 4 is a very small number, so this does not work. It would work if there were many children. or if there were a large number of children. That's why we do genetics experiments on flies and rabbits, because they breed very fast. Now, before we understand this topic of determination of sex in humans, now we have to understand that there is a slight difference between a male cell and a female cell. In the male cell and the female cell, the 22 pairs of chromosomes are the same. They're the same in a male or a female cell. But the 23rd pair, because we know human beings have 23 pairs of chromosomes, in the male cell, one is a little longer and one is a little shorter. So this one is called the X chromosome, and the shorter one is called the Y chromosome. So when in the testes meiosis takes place, the sperms will either contain X chromosome or Y chromosome. So out of 50 million sperms, 25 million will be X and 25 million will be Y chromosomes. So sperms can be different in this way. While the ovum, because the female gender is decided by the 23rd pair, which is XX, which is the same length. So the ovum will always have an X chromosome. Now, if this X sperm fertilizes the X ovum, so we get XX in the zygote. Now, this will develop into a female child. But if the Y sperm fertilizes this and in the zygote we get XY, then this will develop into a male child. So, determination of sex in humans is determined by the sperm. So the sperm, whichever sperm fertilizes, and sperms are in equal numbers. It's not that if a couple has only 4 sons that they have more Y sperms. No, no, no. That's not the condition. Or if a couple has 4 daughters, it doesn't mean that there's something wrong with the sperms. But it's just a matter of chance. It's just a random thing. Just when you throw the dice of the Ludo, sometimes you get 6 in a row. Sometimes you don't get 6, 10 times even. So it's a random thing of having either the X sperm fertilizing it or the Y sperm fertilizing it. Now let's look at the topic of gene mutation. In gene mutation, example is sickle cell anemia. So one gene, this is the gene for hemoglobin, is faulty. Now how is it faulty? There is a change in the base sequence of the DNA. You see, the DNA codes for the different sequence of amino acids. So there is a change in the base sequence of the DNA, and that results in the hemoglobin which is formed has the incorrect sequence of bases. So if it has the incorrect sequence of bases in its gene, but it will have the incorrect sequence of amino acids. So the hemoglobin will have the incorrect sequence of amino acids, but the change will be due to the base sequence of the DNA being faulty. So this is going to result in the hemoglobin formed has an incorrect sequence of amino acids. One amino acid is changed, and this results in the hemoglobin becoming less soluble. So the sickle cell, you see the red blood cells are pulled out of shape and they become sickle cell shaped. The normal one is this, but this is the one with sickle cell anemia. So the person with sickle cell anemia doesn't live very long, because these sickle cells get stuck in the capillaries and they stop the flow of blood and they block capillaries. So part of the body is deprived of glucose and oxygen. So they have severe pain and they have other issues. So people with sickle cell anemia don't live very long, and they have to live with these. There's no treatment for it, sadly. Now, another type of mutation is called the chromosome mutation, and this an example is Down syndrome, in which the change, either there's a change in the chromosome number or there's a change in the chromosome structure. And sometimes what happens is that the ovum may have 24 chromosomes and the sperm will have 23 chromosomes, but the ovaries are churning out faulty ovums, and this has 24 chromosomes. So when that fuses, the zygote has 47 chromosomes. Now, the human being that develops this cell is going to divide. Now, every cell of this human being will have 47 chromosomes. Now, this person that develops, whether it's a boy or a girl, is going to have a disorder which is called Down syndrome, because this extra chromosome is not going to be any helpful. It's going to cause genetic defects in that human being, and that person is suffering from Down syndrome. So it's a genetic disorder, and I'm sorry, again, we do not have any treatment for it. We cannot do anything. This child is born and then it is found out that he or she has Down syndrome. Now, causes of mutation. Mutations occur naturally as accidental changes to DNA or to chromosomes during the cell cycle. The rate of mutation can be increased by some environmental factors called mutagens. Now, the example of that is chemicals in cigarette smoke, sometimes nitrates and nitrite preservatives in hot dogs and other processed meats, benzyl peroxidate, common ingredient in acne products, then infectious agents like the human papilloma virus, HPV, and the bacteria Helicobacter pylori, which is spread through contaminated food. Then exposure to radiation, too much of UV radiation results in skin cancers, X-rays, medical, dental, and other will also cause mutations. Just a recap of all the processes: chemicals, radiation, X-ray, cigarette smoke, tar in cigarette smoke. Now coming to the topic of sources of genetic variation in populations. Number one, mutation. Number two, meiosis, which is responsible for sexual reproduction, and random mating and random fertilization. So these are the 4 points which are absolutely essential for genetic variation in populations. And I mean populations of birds, I mean population of tigers, population of elephants. We can't say population of birds because we have to be name one specific bird. So we say population of green parrots. So you can't say population of birds because birds are different. They're different species of birds. So you can't say population. So population of pigeons. So these the causes why they are why is there variation? Why are not all pigeons alike? Why do the colors vary between? They can be a little gray. They can be white. That's because of mutation, meiosis, random mating, and random fertilization. Mutation is a permanent change in the DNA sequence of a gene. And mutations in a gene's DNA sequence can alter the amino acid sequence of the protein encoded by the gene. Mutations changes in the DNA sequence can result in new traits. Sexual reproduction, new combination of traits can be created in gametes during meiosis. The wide variety of gametes created will join together to make very diverse offsprings. Sources of genetic variation: mutations, change in DNA sequence that does or does not result in a change in the phenotype. Some mutations alter the individual's fitness, some do not. Then gene shuffling, causes of most heritable differences due to meiosis. Crossing over increases genetic variation. We studied this in A levels. This also, let's leave it for A levels. Random gamete fertilized. So 50 million sperms. Which one fertilizes the ovum? Just a random chance, and does not change the gene frequency. This completes the 17.3 component of the chapter on inheritance, and 17.4 will follow in the next video.